Hello everyone, welcome back to Pharma Palette. Today we will be learning about mitosis, the process of cell division with the help of a neat diagram. I will be drawing the each stage on the board, so make sure you note down the details of every phase carefully. So before starting the topic, we have to know what is cell division and mitosis. So initially just assume that this is a parent cell and this is giving rise to two daughter cells. So the cell division is nothing but it is a biological process for producing the two daughter cells from the parent cell for growth, development and reproduction is called as cell division. Let me repeat the definition once again. The biological process of for producing the daughter cells from the parent cell for the growth, development and reproduction is called as cell division. So mitosis is a type of cell division in which a single parent cell divides into two identical daughter cells that will be having same number of chromosomes as that of the parent cell. So that is mitosis. Now let's try to learn the complete mitosis process through diagrams and with some key points. Generally, the mitosis is divided into four phases. So let's discuss each phase in detail. I will be drawing the diagram and also telling you some points. So make sure you note those points and the diagram will be on the screen. So before starting the mitosis process, we have to know about the interphase. So this phase we will be calling it as the late interphase. This part we will be calling it as centrioles and this part is called as centrosome. Inside we will be having nucleolus. The outer covering is called as the nuclear envelope. We can also see the parts like chromatin and the outer part is called as cytosol. The outer membrane is called plasma membrane. So now let's start the first phase which is called as prophase. Now this phase we will be calling it as the prophase. In this we have early phase and also the late phase. What happens in the early phase? In early phase, this part we will be calling it as the chromosome. And on the chromosome, we will be having two structures which is called as chromatids. Now coming to the late phase, this part we will be calling it as kinetochore. It is called as kinetochore microtubule. This part will be calling it as the aster microtubule and this part will be calling it as non-kinetochore microtubule, and there will be also some kind of fragments here. Now these are called as the nuclear envelope fragments. So in the late phase, the kinetochore microtubule, aster microtubule, non-kinetochore microtubule, all the three together they will be forming the mitotic spindle. So the first phase in the mitosis is the prophase. Now what happens in this prophase? There are two stages. One is the earlier one and the next one is late stage. So what happens in the earlier stage? So there is a centrosome. This centrosome will be divided into two parts. So this divided two parts will be moving to either poles of the cell. They will be present in between the kinetophore microtubules. In the early phase only, there will be each chromosome that will be divided into two pairs attached to the centrosome. So in the early phase, each chromosome will be divided into two and those pairs will be attached by the centrosome. And we can also see the presence of chromatin here. So inside we will be having some chromatin. So that chromatin will be useful for condensing into chromosomes. That will be condensing the chromosomes. Why is this condensation needed? Because this will be useful for preventing the entanglement. What is entanglement? Entanglement is simply the overlapping of the DNA. So in the prophase, what all points you will be including in it? First thing is, they will be divided into two parts. They will move to either poles of the cell and you will be drawing this diagram. Then in the early phase, each chromosome will be divided into two pairs and that is attached to the centrome. And there is a chromatin which is present here, and that chromatin is useful for condensing into chromosomes. Why is this condensation needed? Condensation is needed because it will be preventing the entanglement. So what is entanglement? It is the overlapping of DNA. So this will be preventing the overlapping of DNA. That's why the chromatin will condense into chromosomes. Let's get into the next phase. Now the second stage is the metaphase. So what happens in the metaphase stage? So as you can see in this figure, there is no nuclear membrane. So nuclear membrane is absent. That means nuclear membrane will be disappeared in the metaphase stage. And also here we can see that the chromosomes are arranged themselves at the center of the cell and they are attached to the spindles. So in the metaphase, there is also this point. So it is this point we will be calling it as a metaphase plate. So this metaphase plate we will also call it as a midpoint region. So what all points you will include in this metaphase? First thing is this is the second stage of the mitosis. Here the nuclear membrane will be disappeared. Third point is the chromosomes are arranging themselves at the center of the cell. They are attached to the spindles. And there is also presence of a midpoint region which is called as the metaphase plate. So write these points and draw this diagram. That is enough for this phase. So now this phase we will be calling it as the anaphase. So before knowing what is happening in anaphase, I just wanted to give you one clarity. This is chromatid, this is centromere, and this is chromosome. So just have this clarity so that we can understand this easily. The anaphase, just include these points. First thing is, this is the third state of mitosis. So second point is, this causes the splitting and separating of centromere. Also causes the movement of two sister chromatids towards the opposite poles. And third point is after separation, these two sister chromatids they are called as the daughter chromosomes. Fourth point is there will be a kinetochore microtubule elongation. Due to the kinetochore microtubule elongation, there will be increase in the distance between the two separated chromosomes, which you can clearly see in this figure. So this telophase is the final state. So once the chromosome movements get stopped, this phase will be started. There will be reappearance of nuclear membrane and also we can see that the spindles will be getting disappeared and there will be constrictions in the middle of this. So there will be constrictions in the middle and thereby it produces two daughter cells. So this part we can call it as a cleavage furrow. That is about telophase. The telophase, what all points you will be including? First thing is it is the final state of mitosis. And this will be starting once the chromosome movements get stopped. Here there will be nuclear membrane reappearance and the spindles get disappeared and there will be constriction in the middle, thereby it produces two daughter cells. So next this phases will be continued again. And that's the complete diagram and explanation of mitosis. So for your semester exams, just remember drawing the diagram, labeling each phase and writing the key points which I have said in the previous slides. This is more than enough for the answer. If you like this video, don't forget to like, share and subscribe to Pharma Palette. Thanks for watching and see you all in the next video.